Hello everyone, my name is Zoe Farrington. I work in the medical department of a company called Ultragenyx Pharmaceutical, and today I'm going to speak about gene therapy. As a brief introduction, Ultragenyx Pharmaceutical is a Californian-based biotech company with an interest in bringing new products for the treatment of rare and very rare diseases. The company is particularly interested in serious debilitating genetic diseases in 3 key therapeutic areas, and these include bone or endocrine, metabolic, and central nervous system or muscle disorders. The company was founded just over 10 years ago by a physician, Dr. Emil Kakis. He began his work developing an enzyme replacement therapy for MPS type 1 in partnership with the Ryan Foundation. The company is headquartered in Novato in California, however now has offices in the San Francisco Bay Area, Massachusetts, Canada, Europe, and Latin America, and employs over 1,000 people. The company's key focus is the advancement of innovative treatments for rare diseases, in addition to providing access to patients who are in need of therapies and supporting the larger rare disease community in general. This slide provides an overview of the company's approved products and those that are in the pipeline. As you can see, there are 2 approved products, one for MPS 7 and another for long-chain fatty acid oxidation disorders. Products for a number of other disorders are undergoing clinical development currently and are at various stages of development. These include for ornithine transcarbamylase deficiency, glycogen storage disease type 1A, Angelman syndrome, Wilson's disease, creatine transporter deficiency, and glycogen storage disease type 3. So what exactly is gene therapy and how does it work? Gene therapy acts by correcting the mutations that cause disease, and it does this by altering the genes involved. What gene therapy does is that it alters the original genetic information of every living cell and therefore permanently fixes the underlying genetic problem by changing the defective gene. The genetic material is usually delivered by modified viruses that are unable to replicate or cause disease. These viruses are known as viral vectors. The genetic material that is delivered to the cell enables it then to produce a functioning protein. The goal of gene therapy is therefore to achieve a durable expression of a therapeutic gene, also known as a transgene, at a sufficient level to improve or cure the disease with minimal adverse events. So gene therapy involves the transfer of genetic material to a patient to treat a disease. Although the concept has existed for decades, clinical investigations only began in the 1990s. The first study involved a rare immunodeficiency disorder and was run by the NIH in the US. Since then, over 2,500 clinical studies using gene therapy have been initiated in various disease areas, ranging from a variety of monogenic diseases to infectious diseases, neurodegenerative diseases, and cancer. There are several techniques for carrying out gene therapy. These include, first of all, gene replacement therapy. This is used to treat diseases that are caused by a mutation that stops the gene from producing a functioning product, such as a protein. This therapy adds DNA containing a functional version of the lost gene back into the cell. The new gene produces a functioning product at sufficient levels to replace the protein that was originally missing. This is only successful if the effects of the disease are reversible or have not resulted in lasting damage to the body. For example, this can be used to treat loss of function disorders such as cystic fibrosis by introducing a functional copy of the gene to correct the disease. Gene therapy can also be used to suppress expression of a detrimental gene by employing RNA interference or genome editing tools. Gene silencing therapy is suitable for the treatment of diseases which are caused by inappropriate gene activity. The aim of this type of therapy is to introduce a gene whose product either inhibits the expression of another gene or interferes with the activity of the product of another gene. The basis of this therapy is to eliminate the activity of a gene that encourages the growth of disease-related cells. For example, cancer is sometimes the result of the overactivation of an oncogene. This is a gene which stimulates cell growth. So by eliminating the activity of that oncogene through gene inhibition therapy, it is possible to prevent further cell growth and stop the cancer in its tracks. Gain of toxicity mutations are mutations in the gene that result in the acquisition of a new function that interrupts the normal function of the protein and leads to cell toxicity. For example, the mutant protein Huntingtin in Huntington's disease, which leads to the death of neurons and manifestations of the signs and symptoms of Huntington's disease. So gene silencing therapy interrupts or prevents a disease by supplementing the genome with therapeutic genes that target specific aspects of the disease mechanism. This is currently being investigated for the treatment of complex genetic diseases and acquired diseases, for example, heart failure and infectious disease. Let's look now at the key steps in gene therapy. First of all, it's necessary to identify a therapeutic gene and design a vector delivery strategy. Step 2 involves insertion of the therapeutic gene into an appropriate vector. A section of DNA or gene containing instructions for making a useful protein is packaged within a vector. The vector is usually a virus, a bacterium, or a plasmid. A plasmid is a genetic structure in a cell that can replicate independently of the chromosomes. It is typically a small circular strand of DNA in the cytoplasm of a bacterium. Step 3 involves delivery of the therapeutic gene-vector combination to the patient, followed by Step 4, in which the vector delivers the therapeutic gene to the nucleus of the cell. And finally, the delivered gene produces a functional protein or alters the function of the gene. Gene therapy can be delivered using non-viral and viral methods. As we have seen on the previous slide, a vector is a vehicle which is used to bring outside genetic material into a patient's cells. Non-viral methods of gene therapy can include direct injection or transfection of DNA, or delivery using non-viral encapsulation, such as the liposome. Gene therapy can be delivered by viruses that have had their viral DNA engineered or removed. This prevents the virus from replicating and causing disease. You can see from the table here that there are various different types of vector. Liposome, herpes simplex virus, adenovirus have all been used previously and have the advantages of being able to carry large transgenes. The difference between an integrating and a non-integrating vector, as you can see here, is that the integrating vector can be introduced into a precursor or a stem cell so that the gene is then passed to every daughter cell. A non-integrating vector is when the gene is delivered to a long-lived post-mitotic cell. This is a slow-dividing cell, and this can ensure the expression of that gene for the life of the cell. Most gene treatment strategies are now based around the use of two vector types, the lentiviral vector and the adeno-associated viral vector. The lentiviral vector is used for ex vivo transfer, and the adeno-associated viral vector tends to be used for in vivo transfer to post-mitotic cells. Adeno-associated viruses are small single-stranded DNA non-pathogenic viruses. They were first discovered from laboratory adenovirus preparations in the mid-1960s and found in human tissue soon after. They were subsequently cloned into plasmids to enable genetic studies and sequencing of the AAV serotype 2 genome. Since then, adeno-associated viruses have been used in over 100 clinical trials. They target dividing and non-dividing cells and are considered a safer option compared with adenoviruses due to their low immunogenicity and non-pathogenic nature. Adeno-associated viruses engineered for gene therapy replace viral genes with a therapeutic gene needed by a patient. Those engineered for gene therapy are non-integrating. Today, recombinant adeno-associated viruses are the leading platform for in vivo delivery of gene therapy. They do, however, have one major limitation in that they can only be used to carry small to medium-sized transgenes. The genetic material of the adeno-associated virus is replaced with the gene of interest to create what is known as recombinant adeno-associated virus. The recombinant adeno-associated virus crosses the target cell membrane and transports the new gene into the cell nucleus. Recombinant adeno-associated virus is recognized by receptors on the cell membrane, triggering internalization of recombinant AAV into the cell. It is then transported uncoated to the cell nucleus and capsid. The capsid is the protein shell of a viral particle which surrounds its nucleic acid. The single-stranded DNA is then converted to a double strand and forms a circular episome in the cell nucleus. The episome is genetic material within the cell that can replicate independently of the host and in association with a chromosome within which it becomes integrated. There are a number of possible delivery routes for the administration of AAV gene therapies. First of all, ocular administration. Subretinal AAV2 injection has previously been shown to result in an improvement in vision. Central nervous system administration via intraparenchymal, intracerebroventricular, or lumbar delivery is already being used for multiple diseases. There is also a potential for some adeno-associated viruses to cross the blood-brain barrier, which is a considerable advantage for a therapy. Via the lungs, AAV delivery has been given via aerosol, although congestion can limit efficacy. Direct delivery to the heart is already being implemented in clinical trials for the treatment of heart failure. Systemic administration via a peripheral vein is considered to be the best option for monogenic inherited diseases. Patients may need to be screened for neutralizing antibody levels before administration, and re-administration via this method can be difficult. Intramuscular delivery, in that muscle is efficiently transduced by multiple adeno-associated viruses, and the muscle can then produce proteins which can act locally or systemically. Because there is a low cell turnover rate, a long-lasting therapeutic action can be achieved. There are a number of challenges associated with gene therapy. Gene therapy is very expensive to manufacture and difficult to produce at high volumes. As we have seen previously, the recombinant AAV vector is unable to deliver large genes, and as a result, some genetic diseases are more challenging to treat with adeno-associated viral vectors. The patient's immune system may inhibit gene therapy via pre-existing antibodies against a given serotype of AAV, or an immune response to the recombinant AAV treatment, therefore preventing effective readministration. And what about the future of gene therapy? Currently, more than 50 recombinant adeno-associated viruses are in clinical development for a wide variety of genetic diseases. Engineering and novel AAV discovery are likely to provide AAV vectors with specialized functions on demand. Scientists are evaluating whether a treatment offers an improvement, whether it works at the dose that's been given, does it work as a single dose or are repeated doses necessary, does the treatment cause any adverse effects, does it target the right cells, does it help the body produce the right protein, and does the treatment work in all patients or only in some populations, and does it cause an immune reaction? There is hope that these rapidly progressing developments will result in new therapeutic strategies for a number of new indications, and therefore help many more patients whose lives are affected by genetic diseases.